Hi everyone, my name is Sai and I'm a postdoc at SATVI, and today I'll be sharing our plan for a pilot study, a pilot single-cell RNA-seq study that forms part of a broader project looking at the durability of systemic and lung immune correlates of BCG-induced protection. And my aspect focuses on bronchoalveolar lavage or BAL cells, so looking at the lung compartment and the site of MTB infection. And we hope that this may give us some new insights into immune protection against TB. Okay, so just a bit of background first. Despite our many attempts to eradicate tuberculosis disease, it still remains the leading infectious cause of death worldwide, and it places a huge burden on communities. And there are a number of reasons for this, for why TB is particularly challenging. For example, TB presents as a spectrum of disease, and we have limited diagnostic tools, which make it difficult to characterize the disease state and diagnose effectively. And even though we have an approved vaccine, BCG, which we've been using for 100 years at this point, it only offers protection in children, but it isn't effective at preventing infection and disease later on in life. But we still use BCG because the development of a new, more effective vaccine has been hampered largely because we have an incomplete understanding of what protective immunity to TB looks like. And that's really why identifying correlates of protection and gaining deeper insights into the immune responses that mediate control or clearance of MTB infection would be incredibly valuable in guiding both vaccine development and improved diagnostics or therapeutics. So this brings me to a key clinical trial done at SATVI that aimed to assess whether vaccination with either H4IC31 or BCG revaccination could prevent initial or sustained MTB infection. But for this project, I'll be focusing specifically on the BCG revaccination arm and not the H4IC31. So for the study, they enrolled healthy South African adolescents who were HIV negative. They had received BCG at birth and they tested negative for MTB infection using the quantiferin test at baseline. And a quantiferin or QFT is an interferon gamma release assay, or IGRA, that measures interferon gamma production in response to MTB-specific antigens. And it's commonly used as a surrogate marker for infection with MTB. These participants were then followed up for 2 years to monitor for conversion to QFT positivity, which would have indicated new MTB infection. And the results for this trial showed that both vaccines were safe and immunogenic. But even though neither vaccine prevented against initial MTB acquisition, the BCG group showed this 45% protection against sustained QFT conversion. And this was quite exciting because previous studies have shown that reversion, or this transient QFT conversion, might be associated with containment of infection and a lower risk of disease. And it's proposed that a sustained conversion is associated with a higher risk of disease. So these QFT reverters that were identified in this trial may reflect some sort of protected group that can control or clear MTB infection. So that brings me to our aim of the study, where we want to use single-cell RNA-seq to identify immunological features that are unique in BAL cells of QFT reverters compared to the other two groups, so the converters and the non-converters. So for our study workflow, we had quite a few technical considerations to decide on, and I won't have time to go into all of them in detail, but I'm just going to focus on a few of the key ones. So for the single-cell RNA-seq, we decided to use the 3' GEM-X kit and the Chromium from 10X. And we'll also be including site-seq staining for 2 main reasons. The first is to help with annotating cell types. But here we're not using the full total seq B panel. Instead, we'll be using the TB-NK cocktail, which includes key lineage markers for major immune populations to help with the annotation. And the second reason is to include these activation-induced markers, or AIM markers, with the hope of detecting some sort of transcriptomic signature linked to activation. And then we've also included CD206, which is a marker for alveolar macrophages, which is a very important and very abundant cell type when looking at BAL. Then our next decision to make was to hash or not to hash. And one of the benefits, of course, of hashing is that it lets you pull multiple samples before gem generation, which means you can load a lot more cells and then sequence them together as well. So it's super efficient in that sense. But the downside is that hashing reagents can be quite expensive, especially if you're needing to optimize or titrate and troubleshoot them. But for us, our main concern was adding additional washing steps because our BAL cells are quite fragile and we've been losing a lot of cells during the washing steps. But luckily, since the study is part of a larger one, we already have whole genome sequencing data for most of the same participants we'll be profiling with single-cell RNA-seq. And for the rest, the remaining samples where we don't have whole genome sequencing, we're planning to generate bulk RNA-seq. So instead of relying on external barcodes or hashing, we're using our inherent barcodes or SNPs. And by mapping the single-cell data back to a whole genome sequencing or bulk RNA sequence reference, we can genetically demultiplex our pooled samples in that way. So finally, that brings me to our workflow for our pilot, which we're doing in 2 parts, but in parallel. So for the first part focuses on unfractioned BAL, and we have 2 conditions here, NTB lysate and unstim. And after an overnight stimulation, we then pull very carefully because different conditions from the same PID can't be pulled. Otherwise, we wouldn't be able to demultiplex with genotype. And then while this is happening, parallel, the remaining BAL cells from the same PIDs will be sorted to enrich for lymphocytes. And this is mainly because BAL comprises like 90% of alveolar macrophages, and we want to maximize the signal from lymphocytes as well without interfering with the unfractioned BAL too much. So then after we sort these lymphocytes, we'd pull all the PIDs and proceed with site-seq staining. And at this point is where the 2 workflows align. And because sample quality is really important for single-cell RNA-seq, we're using the Levy-cell, which is able to levitate live cells separately from dead and dying cells. So at this point, after enriching for live cells, we'd load all 3 of these pools onto the Levy-cell, to Levy-cell to enrich for live cells and remove the debris. And then finally, we'd load our chip. And because we're multiplexing at this point, we're hoping to recover about 60,000 cells from each pool. So we'd be loading 90 to 100,000 cells in each well, and then generate gems and proceed with the standard 3' GEM-X protocol. So lastly, just as a summary, we're really excited about this work, especially since single-cell RNA-seq is relatively new in this field, with only these few studies so far that have looked at BAL cells and TB disease, and they've all focused on some form of protected group. And this is where we fit in by looking at reverters, which is our protected group of interest. And we really hope that by using single-cell RNA-seq with the breadth and depth of information that it can provide, where it's really a high resolution approach, we're hoping that it'll offer some sort of breakthrough in understanding not just the abundance or quantity of cells present, but also their functionality of the immune cells at the site of infection, to better define lung immune protection. Yeah, just lastly, a huge thank you to everyone involved, the whole SATVI team, and especially the diagnostic team. You guys have been really amazing at helping us set this up. And then the rest of our collaborators and our funders, of course.